Hi, everyone. I really appreciate you having me here. What I'm hoping is, I missed yesterday, but hearing that it was overwhelming, what I'm hoping to do is introduce you to some of the vocabulary that you need to continue talking to scientists and to continue moving your research forward. Please note my email at the bottom. So one of the great things about the company I work for is we're empowered to use some of our time to help people in the rare disease community, regardless if we're working on your disease or not. If you have questions, you just want to know the definition of something, feel free to reach out. It's sometimes easier for one of us to just tell you what the answer is than for you to search for 30 or 40 minutes. So please feel free to do that. And what I'm going to do today, unless I run out of time, is sort of just walk you through what are the therapeutic options that exist and sort of how do they work. And if we have time, I'll leave you with some sort of thoughts to just consider, especially if you're talking to researchers, to really get their opinions, because they'll be the experts on all of these things. So just a quick disclaimer, I'm an employee of Ultragenyx. These opinions are solely mine, do not necessarily reflect the views of Ultragenyx. So if we start just very basically, we're going to talk about gene-based therapies, genetic therapies. What is a gene? So a gene is a basic genetic unit that encodes for a function. And so if you look over here on the left, I don't think I have a pointer, right? If you look over here on the left, you have a cell. Oh, is there a way to do it?
Yeah, don't worry.
Awesome, thanks. I'm really only good at one or two things. I'm glad she could help me. So if you just think about, any part of your body, you're made of cells. Right, no laser pointers. Each cell has instructions to survive, essentially, to do things. And that's contained in what's called a chromosome. So if you look up here on the left, that's basically your DNA all compacted. And to think about how compact it really is, it's quite amazing. It's like sticking 40 kilometers of fine thread into one tennis ball, is how it's actually stuck into each one of your cells. And if you took all the DNA in your body and you laid it end to end, it would actually go to the sun and back 4 times. So there's a crazy amount of DNA that's been compacted into your cells. And so you have to sort of decompress that, right, to actually access it. And so what you do is as you sort of zoom in a little bit more, you get to the actual DNA. And then you have long stretches of DNA. And when you have those stretches, a functional unit of it or a section of it is called a gene. And that's what has the function. And if you zoom in a little bit more, the DNA is actually made of what's called nucleotides, which are essentially letters. And those letters, depending on how they go, encode for functions. It's important to note that every person in this room has mutations throughout their DNA. It's just most of them don't really do anything. But sometimes they do change a function. Another important thing, just to help you sort of speak to researchers, is, as I mentioned, the genes are all over your cell, they're all over chromosomes. They actually have an address. And so the CTNNB1 gene is located on chromosome 3, specifically at 3p22.1, MED13L on chromosome 12, 12q24.21. Not expecting you to memorize that, but it can be very useful, especially if you're interacting with your SAB or something. You can tell them, this is the gene, this is where it is, this is how you find the things that I need you to look at. Okay, so you have the DNA, you have a gene. What actually happens with that? And so the DNA encodes for about 20,000 genes. And most of those have very discrete cellular functions. And it's really what's required for our day-to-day existence, all sorts of things we never even consider. That DNA, this is called the central dogma, is transcribed into mRNA. That is then translated into a protein. And proteins are the things that actually have function for the most part. So examples of protein functions: processing the things that you eat, contracting your muscles, replicating your cells, moving one protein to another part of the cell to help one of the things I just mentioned. And what gene-based therapies aim to do is replace or repair these pathways and the molecules associated with them when they're not providing the typical function. And as I'll walk you through, there's a lot of different strategies to do this. And one of the things I really hope you take away from this is it's not always predictable what's going to work. And you probably saw that yesterday when you were listening to some of the scientists. Things that look like they should work don't always, and things that look like they shouldn't work sometimes do. And so that means that you have to really support and fund research from a variety of viewpoints to really maximize your likelihood of being successful with what you want. So the most basic definition of gene therapy is treating a genetic disease by introducing genetic material. And I'll talk about this in more detail, but again, if you think about sort of, you have the cell, you have the gene, and you're trying to do something here, so the protein that's made has the right function. I'm also happy to share these slides if you guys don't want to take pictures, but you're welcome to take pictures. So to sort of still basic level, what are the different approaches that you can take? So I'm going to talk about gene therapy, I'm going to talk about gene editing, and then I'm going to talk about sort of gene-based therapies like antisense oligonucleotides and things like that. So the most common form of true gene therapy is called gene addition. And so what you have here is if you have what we're going to call a disease cell or a cell that's lost function, what we can do is add back a gene. It then, as you can see here, makes the same but a different version of that protein, and it restores the function. So in other words, you're adding back a functional copy of whatever is either missing or is mutated. In another case, sometimes you actually have too much of a gene. And so as you can see here on the left, if you have either multiple copies or if you have multiple numbers of them being made for a variety of reasons, that can also cause disease, especially in the brain or the central nervous system. There are often diseases that are associated with having too much or too little of a specific protein. And so what you can do is you can inhibit those sequences or those genes, and you can add an inhibitory sequence. We'll talk about that in a few minutes. And if you inhibit some of those, you can get back to what we would consider to be a healthy cell, inhibiting expression to restore a function. And then the last, and definitely, I would say the most exciting, but also the most all over the news in the last probably 10 years, is gene editing. And so what you have there again on the left is a mutated gene. You incorporate this complex that can actually go in, find the specific part of the DNA or the chromosome, and make edits. And you edit that back to a typical copy. And then you should have a healthy cell that's restored function. I'm just going to warn you that the promises in the media are still overstating what this is capable of. It is amazing technology, and it's starting to really work in the clinic, but it's also a long ways away from treating everything. Okay, so that's gene therapy, and we'll talk about that more in a minute. For gene editing, there are a variety of things that you can do, and I just want to familiarize you with these. I'm not expecting you to remember these because even the people that are studying these are constantly updating these. This is one of the most nascent fields right now in biology. So you have this gene editing complex. If you've heard of the word Cas9, that is the actual molecule that goes in and performs this. You can use gene editing to remove DNA, or you can use it to add DNA back in. As I mentioned to you earlier, DNA uses letters, and you can actually go in with specific enzymes and change those letters. And if you have a mutation that's like 1 or 2 letters, sometimes you can use base editing to go in and do that. If you have a lot of mutations or a long stretch, this is not a good approach for that. One of the newest things that's come out is called prime editing. And what prime editing allows you to do is insert longer pieces of DNA than have been possible previously. However, as you can imagine, as you put something more complex in, the efficiency of that is reduced. So this is something that's in clinical trials right now. Those have been started recently. It's incredibly promising technology. It's definitely still being developed. And then the last two are, honestly, these are more common, I think, in experiments in the lab, but theoretically it can be done in a person. You can either use CRISPR or Cas9 to turn on a gene if you have too little of it, or you can turn off a gene if you have too much of it. So I think you can see even just now, these are multiple approaches to sort of do the same thing. have more of a gene in protein or have less of it. And it's just which one of these approaches will work for the biology of the disease that you're trying to treat. Because I mentioned it's not always straightforward. Okay, and then what are gene-based therapeutic approaches? So some of this, by the way, is just semantics. So when you talk to certain people, they're going to have feelings about what one thing is and one is another. So this is my opinion. But again, I'm happy to chat with you about this if things come up. So the ones that I'm going to explain to you now really are more associated with the mRNA state. So not the DNA in the gene and not the protein at the bottom, but the sort of middle part where you need to make the RNA. And if you're missing pieces or you're doing what's called splicing wrong, you actually have to put these pieces together. You can use antisense oligonucleotides or small molecules to affect that, as you can see here. If you have your RNA is being turned over by your cell, the cell for some reason thinks there's a problem with it and it gets rid of it, you can use antisense oligonucleotides and other technologies to stabilize those, to keep them around and get your protein level up. It's important, though, to note, as I've mentioned several times, these different modalities work differently for every disease. And what's going to be really critical is, I think, supporting research that helps you understand the mechanism of disease so you can best pair these different things I've introduced to you with what will actually function in a person. Okay, so how do these things actually work? So I'm just going to walk you through sort of 3 examples quickly. So again, if we think about what I presented in the very beginning, you have a cell. It has all your DNA all packed up. You have genes on your DNA. Those are made into mRNA, made into a protein. And this is actually what a functional protein looks like. They don't all look like this, but this one looks like this is actually one that turns cells green. But they have a 3D structure. They're not just these lines. They really have structures, and that's how they do things and interact with each other and that sort of thing. So if you have a cell that is diseased, then you no longer have this functional protein. If you're going to do gene therapy, so most likely adding the gene back, you put it in this viral capsid. It's like a protein shell. You put it into the cell. You bypass these things, and then you make a replacement protein. And you'll notice it has the same structure as the green one, but I've put it in a different color because it's no longer coming from your DNA. It's coming from DNA we've put into that cell. So it's similar, but from a different source. You can do gene editing, and you can, again, in this pathway, directly affect the DNA, which will then have an effect on your mRNA, protein, et cetera. Or you can use an antisense oligonucleotide or other types of treatments to directly affect the mRNA, either the mRNA itself or whatever's some of the production of that mRNA. So you can see that there's different ways of putting sort of the same ideas into place. And for the DNA and the mRNA component, again, you get a functional protein. In one way, sort of the difference is, though, by going back and doing something directly to your cell, you're sort of restoring the protein that comes from your own body versus putting one in separately. Sort of a simplified way of thinking of it. So this is the introduction to these kind of concepts. I wanted to leave you with some things to think about, to engage your scientific advisory board about. I should note, I am not an expert in either of these diseases, so I'm just giving you my opinion. Definitely talk to the people in your community that are the experts. So for CTNNB1 syndrome, if we think about gene addition therapy, there are some things that you have to sort of check off to see if that will work. In the case of this gene, the coding region is about 2,400 nucleotides or letters. That means it's small enough to fit into this virus shell over here. So that fits like the most basic test of can you do gene addition therapy. But what's really important is understanding the target tissues and cell types that need to be treated. So I think it's really clear that the central nervous system or different parts of the brain and probably the spinal cord need to be treated. I think what's unclear is, do you need to treat muscle? Do you need to treat heart? Are there other types of tissues we're not thinking about that need to be treated? And also, sort of what is enough? And what I mean by that is sometimes don't make perfect the enemy of good enough. If you can do something that really helps symptoms, helps life, sometimes it's worth doing that and working on the additional things later. So I would just encourage you, again, when you're working with your SAB and conceptualizing the research that you want to support, to think about those things. You will likely need to characterize regulatory elements to limit overexpression. And as I mentioned earlier, for a lot of different brain diseases, if you have too much of a protein or too little of a protein, those can both cause diseases. Not always, but often. Okay, if you're going to do an RNA-based therapy, CTNNB1 is autosomal dominant, mostly haploinsufficient. So what that means is you have one copy that's atypical and one copy that's typical. And what you can do, in theory, is go in and upregulate your healthy or typical copy. And that can help offset the other copy that's being generated from. So you could use an antisense oligonucleotide approach to increase the protein that's encoded by this gene, beta-catenin. And not to get into too many details, but there's also an approach where you could target a specific exon, skip that exon. So what you're doing is making a smaller protein, but you're taking a chunk of that protein out that seems to be causing issues. So the real takeaway here is there are multiple options, but they have to be investigated. And then for gene editing, it has huge potential. The potential is to directly fix an underlying genetic cause in that cell. It's important to note that you have to identify a clinically relevant area to edit. If you just do kind of a quick search on both of these diseases, you can see that there's mutations all along the gene, right? And so you have to actually make these sort of bespoke for each mutation. And so your choices are to make a lot of different drugs or try to make a drug that targets the most common area. But regardless, you have to determine what area you're actually going to edit. Although CRISPR is relatively new, there are, I think, at least one approved drug for CRISPR for beta thalassemia and what's the other one? Sickle cell disease. So it's something that the FDA is familiar with. It's something that's really gaining traction, but it's something that a lot of work has to be done to get it to work in the central nervous system. The example I gave you was in blood cells. Those are a lot easier to manipulate than the brain. And it's important, just as with the others, to understand the target tissue, the cell type, and here it's actually important to understand sort of the number of cells or the area of the brain that you need to target. Because for something that's like liver, you can actually do a subset of your cells and sort of make up for it. But if each cell needs to be changed, then that changes how your delivery has to be. So that's something that you really need to work with researchers on, is to understand, do I need to get 20% of the tissue? Do I need to get 100% of the tissue to expect therapeutic benefit? Because that will help you understand the threshold that you need to reach to really be successful in the clinic. Okay, 2 minutes, and I think I'll be good, Rachel. Is she gonna like get the cowbell out at me? Right? Okay, so let's switch to MED13L. For gene addition therapy, this is actually a bit more complicated, although I just heard them say it's possible for both, and I do think it is. It's just going to take more work. So MED13L is actually quite large. It's about 6,600 nucleotides. That's about 2,000 nucleotides or letters too large to fit into AAV. But there are options, and we won't go into all the details, but basically there are a variety of technologies, some of them old, some of them new, that do allow you to put larger genes into cells. That is going to take more research. It has to be determined empirically for each gene and each system that you're working with. And just to be honest with you, most of these are pretty low efficiency. And what we have found as a field is that I think in many times it's harder to work with neurons and other brain cells than, say, again, a liver. And so you just need to be aware that there is going to be a threshold that you have to meet to make this work. That being said, this approach has worked for Duchenne muscular dystrophy, and it's being worked on for other types, not in the clinic, I should mention, in dogs and monkeys and things like that. But there are possibilities. It's just going to take, effort. RNA-based therapies, honestly, probably a better approach, but again, I'm going to suggest that you guys look at everything. This is driven by an individual genetic diagnosis. For haploinsufficiency, you can take a similar approach using an ASO, possibly skipping certain exons depending on what the mutation is. Gene editing might be better for the microdeletions if you could do prime editing, although it also might be better just to do gene addition, but as I mentioned, that could be kind of complicated. Prime editing, although incredibly promising, is just challenging at the time. There are some ongoing sort of new clinical trials, and as you do research, pay attention to how that's going and see if it can be useful to you. 2 more slides, so don't get out the cowbell. I've mentioned these things, but I'm just going to sort of mention them again. Having clear biology is the most important thing, not only to be successful in the lab and in the clinic, but to get a company, if you want a company, to work with you. I would argue if you want to start your own company, it's still really important because it's going to help you get investors, it's going to help you get money, and it's going to help you be successful. And again, invest in multiple modalities if financially feasible. Some work better than others, and a lot of that is disease specific, and it's not always clear ahead of time. And I'm sure some of the scientists told you yesterday, but science is almost never linear. Some people like to show it sort of as a map where, of course, if you wanted to go from here to Austin, you would do a straight line. But what often happens is you go like this to end up in Austin. And so it's really painful because of the gravity of what you're trying to do. But unfortunately, that's the way science works. Okay, I've already driven these home. I'm not going to say them again. Please feel free to reach out to me if you have any questions, and definitely take advantage of Rachel. I'm sure you guys know her, but she's just absolutely amazing and such a great scientist and a really good resource for you. So with that, thank you. Appreciate it.